Oh, thank you very much. Thank you very much for the kind invitation, Igor, to present at this meeting. So what I'll be presenting is using short-chain fatty acids really as a case study of how metabolites produced from gut microbial activity can alter appetite regulation. Yeah, man. There we go. So yeah, short-chain fatty acids are obviously not the only metabolites produced by the gut microbiome, but they have a very important roles. So primarily, you get your short-chain fatty acids produced from carbohydrate fermentation. And their production is primarily determined by the amount of our diet. So why is this potentially important for appetite regulation? And I think that this was nice work from Andrew Reynolds and Jim Mann's group in New Zealand. So this was a large meta-analysis sort of showing the positive impacts of dietary fiber on health outcomes. So at the top, observational studies, so showing that high fiber diets have this really important role in reducing your relative risk of common non-communicable diseases and all-cause mortality. And what they also did as well was take information from randomized controlled trials where they tried to identify mechanisms to explain this effect. And what you can see at the top, it shows this change in body weight that in trials which have a high fiber diet compared with low, you see this reduction in body weight. So it could be that eating more fiber actually has this positive effect of reducing your appetite and energy intake to influence an individual's body weight. So one of the mechanisms why fiber could be good for you is obviously through this production of these short chain fatty acids, so primarily acetate, propionate, and butyrate. And it was around about 15 years ago now, I would say, that there was real interest in sort of short chain fatty acids as potential regulators of appetite. And this was through the identification of the short chain fatty acid receptors, so 3 fatty acid receptor 2 and 3 fatty acid receptor 3, and the fact that these were expressed or co-expressed on the L cell in the colon. And what Igor has just mentioned, it is this L cell within the gut that actually secretes these appetite suppressing hormones, PYY and GP1. So numerous research groups have shown, either through cell models or in this instance in vivo rodent model, that basically if you increase the amount of short chain fatty acid within the gut lumen, you get this increased secretion of PYY and GLP-1. So this was work, say, conducted by Ariana Pasikas at Imperial, showing that if you put more PA, propionic acid, into a wild type mouse within the gut, you get this rapid secretion of both PYY and GLP-1. But if you have your free fatty acid receptor 2 knockout mouse, you no longer see this increase in PYY and GLP-1. So real nice evidence that short-chain fatty acids of local effect within the gut lumen, that they can alter your appetite through the secretion of these appetite suppressing hormones. However, these short-chain fatty acids don't just stay within the gut, they are absorbed within the circulation. So what these numbers show here of acetate in red, propionate in green, and butyrate in purple, as this is data from humans, so individuals undergoing abdominal surgery where they were able to take blood samples from the portal vein, sort of indicative of what's actually coming be absorbed straight from the gut. So you can see that there are, particularly with acetate, large amounts of these short chain fatty acids in the portal vein. And what researchers at the University of Lyon identified is that you have the free fatty acid receptor 3. So one of these receptors for short chain fatty acids is actually expressed within the portal vein. And what they again did in a rodent model is using CFOS imaging, looking at neural activity within the brainstem. If you infuse saline as a control or short-chain fatty acids straight into the portal vein, you see this immediate activity within the brainstem. So there is this direct sort of gut brain signaling via stimulation of receptors within the portal vein. If we return to our slide looking at short-chain fatty acid systemic availability, what these studies did with humans undergoing abdominal surgery was they took paired samples from the peripheral circulation. So what this allows you to do by comparing arterial blood versus portal vein blood is look at how much uptake of short-chain fatty acid is occurring across the liver. And what you'll see is from the reduced numbers in the arterial blood, there is this large increase or absorption of short-chain fatty acid, particularly butyrate and propionate within the liver, where these short-chain fatty acids actually contribute to energy production. So they act as both lipogenic or gluconeogenic substrates. So it could very well be that if you're increasing your short-chain fatty acid production, you are simply reducing your energy intake just from a knock-on effect of basically this increased energy harvest coming from the gut. And finally, another mechanism that how short-chain fatty acids can alter your appetite is via direct effects within the brain. So particularly acetate. So as the previous slide showed, that's really the only short-chain fatty acid that reaches peripheral blood in an appreciable concentration. So this was work conducted by Gary Frost at Imperial. So what they first showed, again in a rodent model, is that if you infuse acetate, an intravenous infusion of acetate compared to saline, is that you see this acute reduction in food intake. And what they then did using a radio-labeled acetate and some appropriate scanning is showed, well, where is this acetate actually taken up and where is it metabolized? So you'll see again that the liver is really the main site of clearance of the acetate. But what they also showed in the top of the scanning image is that the brain is also an important site where acetate is taken up. And what they were able to do is by tracking the metabolic impact of this acetate is that they showed is what you see is an increase in the neuropeptide populations of this POMC neuron, which has this important role in suppressing appetite. So really putting this all together is that what it sort of demonstrates is that some of these metabolites that are being produced by the gut microbiome, they can alter your appetite regulation through these local effects by stimulating the release of your appetite suppressing hormones. However, metabolites do get in the peripheral circulation, and with the case of acetate, it may very well be that by increasing acetate availability, that itself might be acting as an appetite suppressive signal directly. These short-chain fatty acids are also important carbon molecules, again, which can contribute to energy substrate production. And again, this might be sensed neurally to actually alter or suppress your energy intake. And finally, that these receptor mechanisms exist for various metabolites produced by the gut throughout the gastrointestinal tract or within the portal vein. And again, there could be this direct gut neural signaling. So to summarize, dietary fiber as a key substrate for production of short-chain fatty acids, and that short-chain fatty acids can impact appetite regulation via multiple mechanisms. But hopefully this has been just a nice demonstration that when thinking about sort of gut microbial activity and what's being sort of produced by the microbiome, it's far from a simple linear story. These metabolites don't just suppress appetite via a single mechanism. There is potential throughout the body through numerous hormonal, metabolic, and neural signals to actually regulate appetite, which I think is important in this sort of Ozempic and Mounjaro era that we tend to be sort of perhaps too much focused on GLP-1 when that is only just a single player in a large system. Thank you very much.